Hi, my name is Kiersten Shyian. My student number is T00678-685. And today I will be labeling the parts of the eye and the ear, which I have taped up here. Alright, so if we start with the ear, the anatomical features include the pinna, which is this big outer part, as number 1. External auditory canal would be this thing here. So the vertical part would be 2A and the horizontal part would be 2B. The tympanic membrane would be number 3, so it's your eardrum, which is here. Your cochlea is this snail-shaped thing, number 4. So number 4 is the cochlea. The semicircular canals would be labeled as number 6 on here. So these are the semicircular canals. The auditory ossicles would be labeled as number 5. So here you've got your malleus, your incus, and your stapes. You've also got the vestibulocochlear nerve, which kind of branches off the back of the cochlea here. So I've kind of drawn that. That's labeled as number 8. And then the eustachian tube we have down here, which is labeled as number 7. So, and then we have to explain what oral hematomas are. So basically, if an animal is shaking their head vigorously due to some kind of ear infection or other issue, it would break up some of the blood vessels here in the outer pinna area. And then it would cause basically free blood to be trapped in between the skin and the cartilage of the pinna, which would create the oral hematoma. So then if we move on to the eye here, so we have our cornea. So our cornea would be number 3, that is the outer layer here. The pupil would be number 4, so it's the space in between the iris here. Which the iris I've labeled as number 17. The ciliary body would be labeled here as number 9, so it's kind of this part here. The suspensory ligament I've labeled as number 18, so this ligament right in between the lens and the ciliary body. Lens is labeled as number 10. The choroid would be labeled as number 11, so it's this part here. The choroid also contains the tapetum lucidum, which is the shiny part when animals are in the dark. Excuse me. The optic nerve would be found here at number 14, so it's kind of this lovely nerve here. Your optic disc would be found around here, so I will label that as number 20. That's kind of where the blind spot of the eye is. The sclera is the outer hard part of the eye, so it would be number 12. Your vitreous humor would be located in here, so that's labeled as number 16. And then the aqueous humor would be found in, so aqueous humor is number 6, so it would be found kind of in this area. And then if we flip this over, do that there. My apologies. If we do the conjunctiva, so we have our palpebral conjunctiva here, which is right under the eyelids, labeled as number 1. And then the bulbar conjunctiva would be labeled here as number 19, so just kind of over top here, the bulgy part. And now if I flip it over, we would have so this would be the medial canthus, so I will label that as number 4. And over here on the other side would be the lateral canthus, labeled as number 1. The nictitating membrane would be kind of found where number 5 is located. So the third eyelid would kind of be under here, and it kind of goes back and lives in this area. And then the location of the nasolacrimal duct would be, I've kind of drawn it down here. So the duct would run into the nasal cavities. So I labeled that as number 6. And that is the list of features for the eye and the ear. Hi, my name is Kiersten Shyian. My student number is T00678-685, and today this is my workspace. So I've got this diagram here and then the assignment, and my computer is on the desktop. So if we first discuss how muscles make the lens flatten, excuse me, or expand to accommodate vision, this is the part that we are going to look at. So this is the basic accommodation of the eye. So we have our lens here, we have our suspensory ligaments here, our ciliary muscles, our ciliary body is here, ciliary muscles here, suspensory ligaments, and then lens. So if we are looking to see distance, our ciliary muscles will relax, which puts more pressure on these suspensory ligaments, which is in turn going to flatten our lens. So if our lens is flattened, that means we will have better distance vision. The lens changes shape due to the muscles. So if we are looking for close-up vision, then that is when these ciliary muscles will excuse me, kind of relax here, contract here, which will relax the suspensory ligaments, and then we'll kind of keep the lens in its normal, more rounded shape, which is going to allow us to see close up. So that's kind of the accommodation of the eye. And then if we go down here, we can kind of discuss the muscles that make the iris open and close. So we do have so the iris basically has two types of smooth muscle. So it has the radially arranged muscles, and then it has the circular arranged muscles. So this would be a constricted pupil. So that is when our radial muscles are going to relax. Therefore, our circular muscles are going to contract when there's too much light, and then it will contract the pupil so there is not as much light coming in. So this would be kind of a regular pupil. So these are kind of relaxed in the same way, so the pupil is kind of about this size. And then if our pupils were dilated because we're not getting enough light in them, or it's too dark, these radially arranged muscles would be the ones to contract. These circular muscles would be the ones to relax, and then that would open up the pupil to allow more light in. The purpose, for nociception is basically the feeling of pain, or the feeling of the stimulus of pain. So there are four parts to nociception. So first we have transduction, which is where we are creating a stimulus that is being converted to the nerve cells. Then we would have transmission, which is where the painful stimulus would actually travel from the nerves to the spinal cord. We then have modulation, which is where basically the feeling or the pain receptors can change on their way to the brain. So it can either amplify or it can suppress the actual pain that the animal is feeling, and that will be kind of transported to the brain. But the fourth part is the perception. So that's where that's where the nervous impulse actually reaches the brain and the animal is going to feel it. So this, like I said, can be amplified or suppressed after the modulation, but it's really important that we as RVTs, and just in general, we understand how that pain or the nerve impulses work to send pain to the brain, because if you put an animal under anesthesia for surgery, the animal could actually wake up feeling more pain than it originally was thought. So it's really important to understand how the pain pathways work, because we can help then relieve that pain or make things better for the animal by providing different drugs during the surgery, after the surgery, so different drugs and therapies to allow the animal to be more comfortable, especially in animals with long-term or chronic pain. And it just kind of is a little bit better so that we understand how these pain pathways are working and then what we can do to kind of alleviate or reduce the amount of pain the animal is going to feel after it wakes up. Thank you.